All right, so I know when we start going over labs, this is one of the first things I say. I always check RBCs first. And there's a reason for that. And we know that when we're looking at ranges, there's a conventional range and then there's a functional range, right? So when we're looking inside functional ranges, we're looking for early stages of imbalance. We're not doctors. We don't diagnose, treat, cure. If you wear that hat, um, for your day job, or you're working in a medical practice and you do have that hat, that's one thing. But what we're talking about here and what the way I practice, we're not diagnosing. We're looking for um, foundational support. So we're going to be looking at those functional ranges. And they're much smaller, right? So when we see things that are not flagged because they're okay in the conventional range, we're looking at trends. Is this trending this way or trending that way? Are we trending towards dehydration? They probably wouldn't be clinically diagnosed as dehydrated, right? If it's not outside of the conventional range. Um, but we're looking at trends, and there's a reason why when RBCs are elevated, um, we say they're dehydrated. So anything outside of a conventional range, right? Then a traditional allopathic practitioner, doctor, nurse practitioner, whatever, would diagnose and label them as being in a diseased state. So we want to make sure that when we're speaking of this, and this is really important for all you guys preparing for level 3 at SAFM, right? That there's a distinction between things that are outside of conventional ranges that are actually clinically elevated or clinically low, and what we're talking about, which is just a trend in a marker. Everybody got that? I should put up the chat so you guys can, yeah, thumbs up and stuff. And if you have questions, feel free to unmute at any point in time, guys. This is not, you know, not just me talking. I want you guys to really start putting on your, switch on your logical brain and start really thinking through these things. Um, and how explaining this to clients sometimes can be a real sticking point, right? Because their doctor says they're fine. And they come back with all these labs that are fine, and maybe one or two markers are out of range, but that's not enough for a doctor to label them with anything. So we have a lot of people that are walking around symptomatic who aren't getting any help because they're not sick enough, right? They're not out of range enough for them to get labeled.
And Joe, this is where we would say something like subclinically high, right? In the
Right.
language of SAFM.
Well, it's so Jen always said this. She goes, if it's sub, it's lower, right?
Yeah.
Super is, right? We're super or sub, right? So, I mean, you can't really be subclinically high. You'd be super, right? It'd be, she always laughed about that when we were talking about it. Um, but right, it's outside of, it's still inside of a conventional range, but yes, it's trending high or low inside that conventional range.
That's just too many characters for the 500.
I know, right? Yeah. I know they need to come up with a acronym for us, like LIR, like low in range, or HIR, high in range, and just use that. So we can just say it instead of, but we went over this last Friday. I just want to like make sure that we have that these are our key RBC numbers that we're looking at. We're looking at the red blood cell, right? That's a count. We're looking at hemoglobin. That's hematocrit. So you need to know when you're looking at these abbreviations what they mean. And these are, I mean, you don't have to, thank God, right? We don't have to type these out when we're doing a chain. But these are these are what we're looking at and what we're going to be discussing today. So an RBC measures how many red blood cells. A vial of blood is a standardized amount drawn. So when you're looking at it, this is how we can do these comparisons because it is a standard set amount of blood that is drawn. So the RBC measures how many red blood cells. The hemoglobin measures the weight of the molecules of hemoglobin in each red blood cell. Hematocrit is a percentage of the blood that contains RBCs. And, we're going to do some visuals here. I think it's really important for you guys to be able to visualize what you're doing when you're looking at this lab panel. I don't want you to just understand. I don't want you to just memorize. This is where people get into trouble with that. What's it? The book that we have. Oh, the Weatherby. I wanted to say Watson, and I'm like, that is not it. So the Dick and Weatherby book that you all grabbed because, Tracy says, grab this book. It's got a load of information. If you just try and memorize, okay, when this is high, it could be A, B, or C, right? If this is low, it's either going to be, like if albumin globulin ratio is low, it's going to be liver dysfunction or immune activations or other conditions associated with these levels are X, Y, and Z. If that's all you memorize is that chart in there, you're never really going to understand and be able to visualize what's happening inside this person's body. So the best part of the Weatherby book over and over again is that little paragraph in the front that says background, that gives you a definition of what the marker is. Other than that, you're just throwing a whole bunch of stuff up against the wall to see what sticks. And it can be, I was so confused when I started looking at this book and trying to figure out what happened. I think the functional ranges in the back give you a good starting place. But if you start looking at vitamin B12 folate deficiency in the back, could be caused by a high this and a high this, and then you have conflicting things. You've got two that are high, but the two that don't match. And then you're trying to figure it out. This is how you figure it out, by being to actually be able to visualize what are these numbers, what do they mean, and how does this look? If I were to actually be peering through a microscope, what does this blood sample then look like? And that's kind of what we're going to go through today. So, RBC, hemoglobin, hematocrit, they're measures and percentages. These MCV, these are more the quality. And then the RDW is the percent variation in size of all the RBCs. So RDW, right? The red cell distribution width is telling you how the cells vary in size. A perfect RDW of 12 is going to have all the blood cells all the same size, right? So you can think about that. If my RDW is smack dab in the middle of range, then all the red blood cells in that sample are the same. The variation is very small, right? But the minute you start raising that number, the minute the variation starts getting higher, then you're going to say, oh, wait, I'm going to have cells of all kinds. I could have cells of all sizes in here. So what does that mean when you're looking at the whole complete picture, right? You need to know that you're not dealing with a consistent red blood cell. You've got some small ones and all the way up to some really big ones. Any questions on RDW, like that basic concept?
No, but that's an awesome picture. Thank you.
Yeah, I love that. So, if the number is lower, like 11 or something, so does that mean that the majority are smaller, or how do you interpret that?
No,
Okay.
it can go lower and you're really looking for the higher one. The lower one doesn't have that much clinical implication. But when the number starts rising, that's when you need to take notice.
Okay. And
Okay.
then this and maybe we're going to get into this, and unfortunately, I have to drop for another call that's way less interesting than yours. But I have my other job's call. But I guess one more question. So this number, we want to think about like red blood cells that carry oxygen, and red blood cells are used in clotting. Like, is that kind of where you're going with this, or?
Yeah, we're going to look at, what with the other numbers, right? How you can distinguish, is somebody really dehydrated? And, but before we get into the nitty-gritty of actually breaking down a CBC, you need to really understand what's going on. I've had practitioners who don't even know the difference between which markers on the CBC have to do with red blood cells and which ones have to do with white blood cells. It's a very simple, basic concept, but if you haven't come through this work, if you just started as a coach or started with some other background, you may have missed that lesson, right? You may not remember that biology class from high school.
Mhm.
So, when you go back and you say these are the red blood cell numbers on a CBC. These are the ones that are specifically dealing with the red blood cells. And you need to know how we get them, what it looks like, what does it look like if this is high or low. So that's kind of how we're breaking this down. And we're going to go through them all today, in a little bit more depth than we did on the basic one last week.
Thank
So,
you. I'll join the recording, but thank you. Have a
okay.
good weekend. Okay, bye.
Yep. So the first two, the RBC, the hemoglobin, and the hematocrit, determine functional anemia tendency. It answers the question, can these red blood cells carry and deliver oxygen? Okay. The MCV, the MCH, and the MCHC is going to shed light on the possible reasons for that anemic tendency. And if you've followed Brian Walsh at all, he does a lot of blood chemistry, but he talks about, are they being produced? Are they being destroyed? Or is there a loss somewhere? And you're going to be able to kind of determine that based on these three markers. And then the RDW we just covered. So why are RBC? Here's your question. Ding, ding, ding. Let's put your thinking caps on. Why are RBC figures higher for men? Why do we have different numbers here in these? When you look at your CBC and you're looking at these functional ranges, why are male figures higher?
Does it have to do with periods and blood loss, because women are cyclical?
Good thought, but no. It is important, I will say, it is important to know if you have a cycling female and you are looking at their CBC, where they are in their cycle, especially if they have very heavy periods, right? You want to know that. You want to know what's going on. It's not because they're bigger, their blood volume's greater, because you're comparing apples to apples with volume of blood here. So, they're larger, so they might have more volume of blood, but in an equal apples to apples blood volume sample, why are there more red blood cells in a male sample?
Does it have anything to do with their muscle size? And they need more oxygen?
Nope. I know it's a stumper, isn't it? Lauren, did you watch this? Do you have the answer? Randy, you have thoughts?
I've watched it over and over again. And I don't remember asking that question about the men. I thought it was because they're like bigger and, you know,
Something inside men. They have
testosterone.
ding, 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 ding. Winner, winner, chicken dinner. So yes, testosterone. Now if you have a female who, for some reason has PCOS, right? And they have higher androgens, that, you may see an elevated RBC, but I mean, that's rare. I mean, that's not a normal reason for the female to have elevated RBCs. If something, um, I know there was a case once where the woman was, um, her husband was using topical testosterone. And for whatever reason that, you know, rubbed off on her and that had an effect. Um, but that is why men have higher RBCs.
Is there more specific detail?
Um, it has to do with, oops, sorry, it's not what I wanted to do. It has to do with the effect of testosterone on the formation of red blood cells in the bone marrow. And I mean, yes, downstream, right? I mean, think of all the reasons why men have higher testosterone and why that might be a advantage for the male in the species. Traditionally, I mean, we got into a kind of interesting conversation about this when this question was posed, you know, why they would have more red blood cells. But traditionally, they're the ones that are hunting and and doing these higher things. They're the ones that might be getting injured more frequently, right? So some of those things were brought up. I thought it was interesting. So when a little baby blood stem cell is starting, it differentiates into all the different cells that we have. It differentiates, they all start the same. And then on the left here is the formation of a red blood cell, right? And it goes from pro-erythroblast to early erythroblast. B12 and folate help kick out the nucleus, right? Then you get into having a reticulocyte where the heme and the iron get added into a mature red blood cell that can carry and deliver oxygen and pick up waste and carbon dioxide, right? And then those stem cells are also formed into myoblasts, which break up into the different white blood cells, into lymphoblasts, monoblasts, which come down here to all your different white blood cells, and also into the formation of platelets. So I don't know if anyone's ever seen like how all those get made and differentiated, but I thought that was, even with my biology background, I don't remember actually seeing it done in this way. So Hmm. So, when you start with red blood cell production in the bone marrow from that hematopoietic stem cell, and it goes through that process, it goes the first 20 hours from the proerythroblast to the basophilic erythroblast, down to where the nucleus is ejected, takes from 1 to almost 3 days. And then at the edge of the bone, you get the reticulocyte into the full erythrocyte. So early cells are globin filled, and as the globin decreases and heme is bound, the cell starts maturing. And the nucleus gets smaller, and then it gets ejected, and then a fully matured red blood cell contains no nucleus.
And Joe, you said it was B12 and folate that are involved in the ejection of the nucleus?
Right. So yes, because then when you're looking at a macrocytic cell and you have elevated MCV, you want to look for this deficiency. Red blood cell production starts here, but they are required to get that nuclear maturation. And if B12 or folate isn't there, the nucleus doesn't shrink. And then the cell is larger than normal. Well, it's macrocytic. You'll see macrocytic cells that are very large in cells, and they're hyperchromic. They'll be very red in color.
I mean, if there's a lot of sites,
I'm sorry, if there's a lot of,
reticulocytes, like more than there should be, why would that?
they haven't figured, they haven't fully matured to the last stage in the red blood cell development, right? So they're stuck here. So,
So that's because of the nutrients.
mhm,
Less nutrients.
right. And if we go to the next cell, right? If you have low hemoglobin, can look for B6, zinc, or iron deficiency. So for heme to be created, you need B6 and zinc. The polyporphyrin ring also needs to have an iron attached to finish creating heme, which can now attach to the globin to make hemoglobin. And without those three nutrients, heme isn't made, and therefore no hemoglobin is made. And so the reticulocyte ends up being small and microcytic. It lacks the oxygen binding properties, and it's usually very pale or hypochromic. So if you're lacking nutrients, you're never going to finish that process. The cells are going to be the red blood cells that you have are going to come out into the bloodstream, and they're going to be somewhere between a perfect red blood cell and one of these ones on these two slides. So when you're looking at the MCV, MCH and MCHC, perfect ones are going to be right here, right? If your mean corpuscular volume is low, you're looking at tiny cells. If it's large, you're looking for larger cells. If the MCH, right, the amount of hemoglobin in the cell is low, they're going to be pale. Or they're going to be very red, right? If there's more. And then the same thing with the MCHC. You're going to be looking for lower amounts of hemoglobin. They're going to be small, they're going to be pale. And over here, you're going to have large hypochromic. Does that make sense? Or is that a good visual for you guys to like actually have an idea of like what are these cells looking like? Yes, no?
Yes.
Okay. Well, I had my chat turned off, Lee. Yes. What is going on here? What did I do? All right, hang on. I have no idea what this is supposed to be. There, let's just skip that cell. So when you centrifuge whole blood, I think, hang on. I'm going to stop share for a second and see what the heck I did to my presentation. I was trying to make this happen today, and obviously, something is going on with slide 15, and I do not know what. There we go. I just have that picture over it. All right, back to presenting. There we are, back to sharing my screen. Ta-da! Okay, back at it. Can you guys see it?
Yep.
Okay.
All good.
All right. So each vial of blood is drawn to a specific amount, and each vial contains both plasma, white blood cells, and red blood cells. So when you get the little vial drawn, right, you guys have all had blood taken. It's filled to this very specific amount. The CBC vial is designed so it holds a specific amount. When they take the whole blood and they put it into the centrifuge, then you have this sample that comes out the other end. 45% of it is going to be the formed elements, the red blood cells, the white blood cells, and then the platelets. You're going to have this little buffy coat here in the middle. And then over here, you're going to have the plasma. So this part is the red blood cells. This little buffy coat is the white blood cells and the platelets. And then your plasma, which transports the material in the blood. Have any of you worked in a lab, done CBC, done any work? Yes, no? So I did. I did an internship. I worked at the VA. I drew blood. And these little vials come out, they go into the centrifuge, they get popped into a machine that does all the work, and then it spits out your CBC. I mean, it's only $8 for a reason, because literally a machine does the whole thing. So plasma is going to be the largest part of the sample. It makes up more than half, about 55% of a regular hydrated perfect blood sample. Right? It's over 91% water. So it's mostly water with a little bit of stuff in it. When it's separated, it's a light yellow liquid. It carries water, salts, lipids, and enzymes. The main role of the plasma is to transport nutrients, hormones, and proteins to the part of the body that needs it. Plasma is the vehicle for everything. Cells also put their waste products into the plasma, so then the plasma helps remove this waste from the body. Blood plasma also carries all parts of the blood through your circulatory system. Along with water, salt, and enzymes, plasma also contains important components. These include antibodies, clotting factors, and the proteins albumin and fibrinogen. So plasma proteins are the abundant substances in the plasma and are present in three major types: the albumins, the globulins, and the fibrinogen, and they play very specialized roles. It's really important that you guys understand, like when you're looking at a marker, it's one thing to just go, oh yeah, that's a protein. Oh yeah, that's a mineral. But do you really truly understand what each thing does? Right? That's where Weatherby's going to give you that little background on it, but you really need to understand what's albumin. So albumin helps maintain the colloid osmotic pressure of the blood. It is the smallest size among the plasma proteins, but makes up the largest percentage. The colloid osmotic pressure of the blood is important in maintaining a balance between the water inside the blood and in the tissue fluid around the cells. When the plasma proteins are deficient, the water in the plasma seeps out into the space around the blood vessels and may result in interstitial edema, a feature of liver disorders, kidney disease, malnutrition, as for examples. And albumin also helps transport many substances such as drugs, hormones, and fatty acids in your blood. Globulins are three types. You have alpha globulins, beta, and gamma. From the smallest to the largest, gamma globulins are your antibodies. So when you get IgG testing for for things, that's the gamma globulins. Alpha globulins include the high-density lipoproteins, HDL, which are important in carrying fats to the cells for building various substances, as well as for energy metabolism. HDL is best known for its role in preventing plaque formation by keeping cholesterol in transport within the blood. And then LDLs are beta globulins, which transport fat to the cells for steroid and membrane synthesis. It also promotes cholesterol plaque formation, which is a risk factor for arterial and heart disease. Antibodies or gamma globulins are also called immunoglobulins. They're produced by the B lymphocytes, a subset of the immune cells. Antibodies are responsible for the body's humoral immune function, recognizing pathogens via specific receptors and neutralizing them by various methods. So this right here is one of the electrophoresis, so you can like split out and figure out which ones are high and which ones are low. It's a fairly cheap test, but I don't know that it's run very often. Colloid is like colloidal silver is a very specific suspension in chemistry. So I mean I can certainly Google the exact definition. Making me pull out my biochemistry brain. So here we go. It's a homogeneous, non-crystalline substance consisting of large molecules of one substance dispersed through a second substance. So think about gels, emulsions. Particles don't settle, and they cannot be separated out by ordinary filtering or centrifuging. So very special solution that our body uses. Is that enough? Yeah, it's used a lot. Like colloidal silver it's what puts that metal in solution, right? Because you would think that it would just be separated out, but it is not. All right. Fibrinogen is an important soluble plasma clotting factor precursor, which is converted into a thread-like protein called fibrin on contact with a sticky surface. The fibrin threads formed in this way trap platelets to form the primary platelet clot on which a stable blood clot is formed by the process of coagulation. So if you have high values, that can be caused by many conditions. So some of the most common reasons that you would have high values, high albumin usually is a sign of dehydration. So you can use that to confirm in your, in your lab work, is this a dehydrated sample? High alpha-1 globulin is infection or inflammation. Sorry, high alpha-2 globulin is inflammation or kidney disease. High beta globulin could be very high cholesterol, low iron, or iron deficiency anemia. High gamma globulins could be inflammation, infection, liver disease, and sometimes some forms of cancer. Low albumin can be caused by many conditions. So some of the most common are poor nutrition, inflammation, liver disease, kidney disease. Alpha-1 globulin, if that's low, it could also be inflammation or liver disease. And it's hard to determine, right? Because some of these, well, it's high for this and it's low for that. Like kidney disease, these things are on both sides, right? Low beta globulin could be poor nutrition. Alpha-2 globulin could be thyroid problems, and low gamma globulins could be problems with the immune system. So if you have a fully hydrated blood sample, right, it's going to kind of look like this. But then as you get dehydrated, this is 91% water. So as you get dehydrated, what's going to happen is there's just going to be less water in the sample. So you're going to have more RBCs because there's less water here. So the blood is going, right? It's going to appear to be higher. Is that a good visual for you guys when you're looking at, when you start talking about the numbers, right? So the only 99 times out of 999 times out of 100, right? Out of 1,000. The reason RBCs are going to be elevated is because there's just not enough water. You're going to fill up this tube with the same volume, right? It's going to look like, let's go a couple back here, right? It's going to look like this in both of the samples, right? But then when you centrifuge it down, and you break it down, your plasma is going to be less because there's not as much water in it. So it's going to appear this way once you do it. When the water intake is too low, several changes will also take place within the body to respond to the conditions. For example, your body's going to increase the production of cholesterol so it can keep the cell membranes pliable and moist. When your body lacks fluids, a series of changes can take place. The increase of blood cholesterol levels can be considered as a perfect example for a change that can happen within your body. The body's going to then try to deposit the increased cholesterol levels within the cell membranes of your body. This is going to be done as a natural protective mechanism so that the body can make sure no more water is lost. This fact has been verified by scientific studies as well. So lack of water, through the view of hemoconcentration, also makes the amount of cholesterol in the blood higher than what it may be.